Thank you so much for organizing the event and inviting me. Man, this is a big room. I'm already nervous. Okay, so today I'll talk about how we use topological data analysis to quantify brain dynamics in individuals with Fragile X syndrome. And I did it somehow. Did I press the wrong button? Okay, somebody's helping us. Okay, who's jet lagged? I am, so bear with me. So the quick outline is we'll talk very briefly about why should we study brain dynamics. By now you already know why we should, and that's why probably you're in the room as well. And then some gaps in our current understanding, and then I'll go to introduce our topological data analysis-based approach on how we study the changes in brain activation patterns, and then a quick validation in task fMRI data, followed by application to resting state data in Fragile X syndrome. Okay, so of course studying brain dynamics is important because it's understanding how brain adapts from one task to the next is basically the vital to understanding cognition as a whole. Plus we know that by averaging data over space and time, we might be losing out on special potential biomarkers for, let's say, ADHD versus depression. So that's my understanding of ADHD versus depression, by the way. Don't tell anybody. Anyways. Okay, so there are many approaches to studying brain dynamics already, as Vince and other people mentioned. And they're mostly divided into two camps. Not that they fight with each other, but just two different ways. One is sliding window-based approach. The other one is more event-driven approaches. In one side, you look at brain dynamics as a continuously evolving temporal dynamics. On the other side, you look at certain sparse events, short events, and then you try to make sense of that. That way, both are amazing and useful in many ways. But there are certain issues that I think still remain, and Vince talked about some of them. For example, how should you choose the right optimal temporal spatial scale in case of sliding window, or best threshold in terms of the other time-based events like CAPS. And also, whether should we model the dynamical landscape of the brain as a continuous manifold versus a discrete. And in extreme case, what we really want to have is some kind of visualization that a clinician can use in an office when he or she sees a patient, where they can actually look at how the brain parses this dynamical landscape and whether they can then use that information in some way to prescribe a treatment. So we basically need some kind of data-driven solution, and perhaps it might be better to take all the points rather than averaging or picking up only cool points, and maybe estimating the underlying manifold would be an interesting way to at least see what's happening there. So to do that, we use topological data analysis, specifically something called MAPPR, which is essentially a fancy way of saying that we are estimating a low-dimensional embedding or manifold as a graph, and this is how we do it. So if I use a toy data set, which is this three-dimensional trefoil knot, essentially, and the first stage is very similar to any of the, if you've ever done dimensionality reduction, you get to these, any two dimensions. From three-dimensional, we go to two-dimension, try to capture some aspect of the data. Then most of the machine learning or unsupervised machine learning will stop there, but then we go a couple of steps further because we want to create a graph out of this. So we bin this two-dimensional space into two-dimensional overlapping bins, and then we do partial clustering. So we essentially want to compress the representation, but not by averaging, but just to compress the representation itself. We cluster the points within each of these bins to create those bigger nodes. And then, because there is overlapping between the bins, whichever nodes then share the points get connected by an edge. So essentially what we do is we go from the three-dimensional data to a graph construction. But something interesting happens here, and that is that when we cluster these points within a bin, we cluster them in the original space. And that's very important to understand because whenever we do dimensionality reduction, we lose some information because you're going from high dimension to low dimension. But by keeping the clustering in the high dimensional space, we can actually capture some information that was lost when you did the low dimensional mapping. So just to make that point one more time, so if you take the same data again and do like PCA or TSNE, and if you look at that square where there is an overlap between the green and the blue and red lines, you will realize that it's actually like a high fly over, the green kind of goes above blue and red. That kind of completely we miss if we do PC or t-SNE and just live there. But because we do partial clustering in mapper in the high dimensional space, I don't know if you can see it, but the green is still kind of above blue and red in this shape graph. And essentially, we actually capture the entire shape, which is actually supposed to be a loop using mapper. So that's where mapper has its advantage. So when you apply this to fMRI data, you essentially take the entire data set without averaging in space or time, so time by space, and then first step is filtering, so you basically create a neighborhood graph. So each node is the entire brain at any one time point. We bin the entire space. We do partial clustering within each bin, and then each basically bigger node is representing more TRs, but not necessarily averaging the data, and then we create a graph out of it. So when we apply it first to a task fMRI data, just to see whether it works, and this is data from Peter Banatini's lab, where they collected a bunch of quick, short tasks, 3-minute-long tasks, as a part of a big continuous multitask experiment. A good thing here is we know precisely when the transitions are supposed to happen, when it's supposed to go from one task to the next. And we also know the behavior at every single step, so we try to predict that. So this is how it looks like. So you take any one person's data, about 1,000 frames, and then this is how the shape graph looks like for that person. Now it's grayscale, but because I know exactly which task that person is doing at every single TR, I can color the graph based on the different tasks, the resting state, working memory, video, and math. And each node in this graph is essentially a bunch of time frames, the whole head time frames. And if they're connected or within the node, the spatial activity is very, very similar. That's why they're connected. So if you look at the graph again, this basically has this core periphery structure, meaning the TRs during the task which require cognitive load, for example, working memory or math, where your brain activation is probably very, very stable because you're going from, for example, doing an n-back memory task, you're really focused, then you end up becoming part of the core, whereas resting state, your mind is wandering all over the place, and you are in the periphery. So that's just a zoomed-in version of the core. We did a pie chart visualization, so we don't want to lose any information. Okay, things get very interesting when you look at shape graphs. We call them shape graphs of different people. So quick quiz time. So one of them is the best performer across all tasks. One of them is the worst performer. So how many people think left one is the best performer? Great. How many think right one is the best performer? So right wins. Not that I'm a conservative, but turns out if you look closely, this person has a lot of pie. The task is represented as pie charts, meaning that a lot of their tasks, there is no task-specific brain activation, meaning irrespective of which task they're doing, they're using similar brain spatial activation patterns, whereas if you look here, there's a very beautiful modular structure, meaning their brain activation during different tasks is a very special task-specific activation. Makes some sense? So because of this modularity, they end up performing better. And if you look at you can associate the simple modularity of the shape graph by behavior nicely. I probably don't have time to go into this, but just by traversing the mapper graph, we can actually extract the transition between different brain tasks at the level of single TR. Given this is, by the way, average over all subjects, so you see individual variations in each subject's data. But on average, without knowing anything about the task structure, we're able to capture changes in brain activation pattern at the level of single TR. So, and you can. I talked initially about it'll be cool to actually look at how you traverse the entire brain landscape, and this is just a movie showing that. So this person starts with the resting state data, and they're going to resting state, just going from one TR to the next, and you get corresponding brain activation, deactivation maps. So now they go to working memories, they'll stay in the core. So it's a data-driven solution. We don't average in space or time at the outset. And we try to estimate the manifold, and then we see how we traverse it to make sense of the data. By the way, the tool is public now. You can play with it. We lovingly call it Dinosaur. If you go to the website, you can check it out. Okay, so now applying it to Fragile X syndrome, and more importantly, the resting state data. So Fragile X syndrome is a very special neurodevelopmental disorder. It mostly affects males. We only have one X chromosome. And it is known as the only genetic cause of autism. So many patients with Fragile X syndrome show symptoms of autism. So how should we apply mapper to resting state data? So the first piece is simple. We again take time by space, apply mapper, create the shape graph. But in the task fMRI data, I was able to color the shape graph based on the task they were doing. In this case, we don't have that luxury. So here we basically do a community detection. Because this is a graph, you can treat it as, you can just calculate communities on the shape graph, and then color the graph based on which community they belong to. And then you can flatten out this compressed representation back in time at the original TR level. And there you get to see basically a sequence of states, like Tatiana also mentioned. Using HMM, they did that. Here we're using shape graph to create these sequence of states. And then essentially we just measure simply the frequency of transition from one state to the next, and you create some Markov chain graph. So again, you have, in this case, we only had about 8 minutes of resting state per person, about 240 volumes in surface space. And this is how the shape graphs look like. And for resting state, and they're colored, in this case, not by the task, but by the different communities that the Louvain algorithm picked up. And from this, we can then create these Markov chain graphs to say, assuming each of these communities are actually some form of state, and then what's the transition probability to go from one to the next. So it's a very small data set. Take it with a grain of salt. We're still validating this approach in human connectome data set. But here, just showing you the 12 subjects with resting state from fMRI, from Fragile X syndrome. And some of the subjects have very dense transition probabilities and densely connected degree of these Markov chains. Some of them have very sparse connectivity in this state space. And it turns out if you just look at this degree of connectivity within the Markov chain, we were able to kind of sort of associate that with autism symptom severity scale, SCQ. So the higher the degree means that there is high likelihood of transition from one state to the next. And lower degree basically means that there is lack of such transition and more repetition perhaps in these dynamics. So very preliminary results, we suggest that lack of transition in these brain dynamic states is associated with higher autism-like symptoms, which kind of makes some sense because that's essentially repetition in behavior, and we're seeing some repetition in dynamics. So that's very preliminary. Again, we are modeling this in large-scale human connectome data. Probably I'll have results next time I see you. And then we have just colored these by male and female just to show you that effect kind of sort of is there in both cases. So this is very densely connected versus sparsely connected up there. And that's it. So thank the collaborators, especially Scott Hall, who has generously given us the data, the funding agencies, our team. And then we have some more applications of TDA on some of the data sets that I welcome you to come. Thank you so much.